Hello students, this is the fifth experiment, that is to study meiosis in onion bud cells or grasshopper testis through permanent slides, and you can complete your writing part of the experiment through this video. Write objective of the experiment: to study meiosis in onion bud cells or in grasshopper testis through permanent slides. Then write the requirements. We require permanent slides of different stages of meiosis in onion bud cells or in grasshopper testes, and also we need a compound microscope to watch these slides. Write the theory part of the experiment. Meiosis is a type of cell division in which number of chromosomes are reduced to half in daughter cells. It occurs in cells where gametes are formed in sexually reproducing organisms. Meiosis is completed in two phases, that is Meiosis I and Meiosis II. Meiosis I is a reductional division, while Meiosis II is a equational division. Meiosis I results in reduction of chromosome number to half, that is from 2n to n, while Meiosis II maintains the number of chromosomes. Write the steps involved in the procedure. First, fix the permanent slide under the microscope. Second, first observe the slide under the low power and then under high power of microscope. After setting the slides, write observations. Under the high power of microscope, following stages of meiosis are distinctly observed. Start कीजिए with meiosis I, prophase I. Prophase I of meiosis I is of long duration and has 5 substages. First substage of prophase I is leptotin. See the diagram. Chromatin fibers condense हो जाते हैं and form thick structures called chromosomes. यहाँ पर सेल में 4 chromosomes शो किए गए हैं. It means we are starting with a cell having 4 chromosomes. Also, nuclear membrane and nucleolus both are distinct at this stage. Second substage is zygotine. Homologous chromosomes form pairs, and these pairs are called bivalent, and pairing of homologous chromosome is called as synapses. The individual of a pair, they are very similar in length and in position of their centromere. Pectine, next substage of prophase I. Now see the diagram. The 2 chromatids of each chromosome become visible. Chromosome के chromatids visible हो जाते हैं at pectine stage, so that a bivalent becomes a tetrad. यह tetrad stage भी कहलाती है. Also, crossing over takes place between non-sister chromatids of homologous chromosomes, means exchange of chromatid segment or genetic material takes place between homologous chromosomes in pectine stage. Diplotin stage. Here the 2 chromosomes of each bivalent move away and homologous are held together at one or more points called chiasmata. You can see chiasmata in the diagram. Last stage of prophase I is diakinesis. Here homologous chromosomes appear thick, nucleolus and nuclear membrane disappear हो जाते हैं and spindle begins to be formed. After prophase I, next stage is metaphase I. Here the bivalent, or you can say homologous chromosomes arrange themselves at the equator of the spindle. Homologous chromosomes spindle fiber के center पर आकर arrange हो जाते हैं and the spindle get attached to the centromere of the chromosome. Anaphase I, see the diagram. 2 chromosomes of each bivalent move to opposite pole and each pole has half the number of chromosomes with 2 chromatids each. Homologous chromosome का separation anaphase I stage में हो जाता है. Now, the last stage of meiosis I is telophase I. The chromosome at each pole uncoil and nucleolus and nuclear membrane reappear. Cytokinesis occurs to form two daughter cells. So, at the end of meiosis I, दोनों daughter cells में two chromosomes present हैं, which is half of that of parent cell. Next come to meiosis II. It includes 4 stages: prophase II, metaphase II, anaphase II and telophase II. In prophase II, chromosomes of daughter cells begin to condense and thick, and nuclear membrane and nucleolus begin to disappear. In metaphase II, chromosomes are arranged on the equator of the spindle, and each chromosome is attached to the spindle by its centromere. Anaphase II, the sister chromatids of each chromosome separate and migrate towards the opposite poles. As a result, each pole receives haploid number of chromosomes. And in telophase II, the chromosomes begin to uncoil and become thin, and the nuclear envelope and nucleolus are reconstituted, and at the end of telophase II, cytokinesis occurs and 4 haploid daughter cells are formed. That is, each daughter cell is having 2 chromosomes. Right precautions to be taken. First focus the slide under the low power and then under high power of the microscope. Second, use fine adjustment while focusing the slide under high power of microscope. So that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.